cells are trying to do is to decrease the level of alpha ketoglutarate, because high level of alpha ketoglutarate will not let HIF become active. If HIF is not active, the cells will not be able to synthesize new blood vessels. And if they cannot do it, if they cannot increase the level of VEGF, they will not be able to move out of the primary organ, and they will not be able to spread. If cancer cells don't spread, they can be very easily treated, right? So with this, I will stop this session, and I will switch on to the tutorial, some of the omics that we do in order to identify prostate cancer drivers. Dr. Aziz, shall we take a 5-minute break before moving into this, or do you want me to continue? Okay. If no one wants a break, then we will take a break anyway after 15 minutes.
Yeah. Can I have a question?
Yes, please, Dr. Aziz.
How are you?
I am very well, except my voice.
Okay. So I was just wondering about the reports where the use of VEGF inhibitors made the cancer more aggressive. So it is considered as a double-edged sword, which can, if you cut down the supply by using the VEGF inhibitors, they find alternate ways to survive, and that is even more aggressive and disastrous.
You are absolutely right. So there's a group, I think Rakesh Jain's group in Harvard. They made those inhibitors, and that is what I think cancer laughs at us, because anything we throw, it always find alternate pathways to grow. So even with these inhibitors, I would imagine many of these inhibitors are orally active drugs, and the way they are metabolized in the body may be very different. Of course, there is a window of opportunity. So perhaps the reason could be that we are not able to we are only achieving suboptimal levels of the inhibition that may actually trigger other pathways becoming active, which will drive cancer, which were otherwise missing. So you're absolutely right. But to really pinpoint what is happening here, I don't have an answer. If you can shed more light, that would be great.
Actually, that's a very genuine concern, because even in prostate, there is standard guidelines to wait and watch for some time.
You're absolutely right. So if you have, so the way prostate cancer is staged is a staging called histological staging, or a Gleason scoring. It was after the name of a scientist from Canada, Gleason. But those people who are put under the watch are the ones whose tumors are not poorly differentiated. If you have very high Gleason score, meaning your disease is very likely to turn really, really nasty, there is no differentiation left. It is highly poorly differentiated carcinoma. Those patients will not be put in wait and watch. So if you have, for example, Gleason 3, Gleason 4 disease, there will be different interventions. So either those patients will be put under active surveillance or watchful waiting, sorry. So there are two things that are done to patients. If the tumor is really intermediate, then they will be put under active surveillance. If they're really low volume tumors, I should point out that many of these prostate cancer patients who they just give away donate their bodies to cancer research. We find cases of incidental diagnosis, meaning they had cancer, which didn't say anything. They died of causes other than cancer, didn't say anything to them. So those are probably low volume diseases. But on the other hand, we have a marker called PSA, prostate specific antigen. If the values of prostate cancer antigen, for example, is normally it is under 1 nanogram per ml of blood. But if you find that the initial diagnosis, you have a value of 20 nanogram per ml, it is very likely that the cancer has started to spread. And that is a very small test. These are the sort of tests that we lack currently. Very minimally invasive. You can just take out the blood. Nobody minds giving some blood. especially if it can save them from any harm caused by cancer. But again, those patients will have likely developed cancer, and the values goes in like thousands
Okay.
sometimes of this piece. So that would depend what you want to treat. I would suggest what Sandeepan was saying: eat less, do intermittent fasting, and enhance your
Mhm.
lifespan when you are healthy.
Thank you. Thank you. I do that often, 16 hours or
Monday and
more.
Thursdays. Yeah, I think we should all do, if possible, who are fit, all intermittent fasting is a good thing. Any other questions before I go? I can discuss still
Alhamdulillah.
those points that I was discussing.
Yeah, please. I'm sorry to keep you.
No, no, that's
And
fine.
we'll
I think
reconvene
the
it
whole point
in
is,
10 minutes.
yeah, 10 minutes is good. So, yeah, in 10 minutes. The rest of you, if you would like, you can just type your questions, please, or we can discuss your questions after 10 minutes. Thank you. I guess we can start now.
Yes, sir.
Great, thank you. I think there is a comment or a question from Sandeepan I'll take before I start. So it says, seems a dead end for fighting against the resilience of cancer cells. As an aspiring cancer researcher, it feels overwhelming how to combat these challenges at times. Uh, how would you advise me to look forward to this journey to navigate the solution for cancer, in particular strategies and aspects in this field? Uh, that's what exactly what I'm going to discuss in this tutorial. Uh, Sandeep and Dr. Aziz can join this conversation by all means. Uh, when we are talking about cancer, it's not something that happens overnight. It is a manifestation of a very long journey of cells, daughter cells having mutations, again, the same cells having some other mutations, making them fitter to carry out these journeys. So they are in a way, as Dr. Aziz excellently puts, they have many targets which are moving. So they are continuously changing flexibly what they rely on in order for them to grow and progress. Um, so there will always be a debate whether we should try or not. I think for economies like economies, the question is, are we able to give our population the therapies which are already available to the wider public through private healthcare systems? If we are not able to give them that, then you also have the fibromuscular part of the gland. So you have fibrotic tissue within the uh, prostate gland as well as the muscular part. Uh, you can also have expansion of these parts, and that is often the case when you see someone who says, "Oh, my prostate hasn't become enlarged." Most of the time, it is not cancer. What they're talking about is a condition called benign prostatic hyperplasia. Now, benign prostatic hyperplasia is not cancer. It can be treated very easily. One of the treatments includes giving drugs due to steroid, but also patients in which the whole gland can be removed. So, when the diagnosis is made at presentation, you will have more than half of the population where the cancer diagnosis is made when the cancer is localized. And this is what Dr. Aziz was telling. These patients may be put on active surveillance or watchful waiting. They may not need a surgery depending on the how extensive the tumor is. Is this just one core? Are there multiple cores? That will also help uh, make a decision on the clinical course for that particular patient. The good thing is if the cancer is identified, diagnosed at this stage, the treatment is super well. And you can see, at least in UK, US, 100% patients survive 5 years after the initial diagnosis, if the diagnosis was made at this stage. So one of the things, as we've been discussing, sorry about that. One of the things we are discussing, cancer tries to invade, and we know cancer invades into the nearby organs. Does it need to metastasize? Metastasis is a different thing. It needs the supply to the blood vessels, to the blood circulation system in order to metastasize to distant organs. Always remember, uh, invasion is local and metastasis is distant. So in this case, you can see this prostate cancer is trying to go into the rectum. So the distal part of the colon, the large intestine, is also trying to invade into the bladder as well. You can see. So this is invasion. This is called locally advanced disease, meaning it is advanced compared to this one. And you will find this in many cancers. You will have cancer, which is a primary cancer. Uh, Patients may have diagnosis of locally advanced disease, which a prostate cancer is a very good example of. And what is happening here is that you see that there is, although local, it has not spread, it has not metastasized, but it is still advanced, meaning it has left the capsule. So prostate gland, the cancer is still encapsulated in this stage. In this stage, what happens is this is the advanced stage of the disease. And what happens here, if the cancer is diagnosed at this stage, of course, the chances of survival are far, far lesser. As you can see, this is a stage 4 cancer, meaning, and this is true for many other cancers, stage 4 would simply mean that the cancer has metastasized to distant organs. So this is a particular topic of interest in my lab, where we study the advanced prostate cancer using a variety of models. We also generate our own models to study what happens in this particular subtype of prostate cancer, which is lethal if it is not treated. So the way the cancer is treated, localized cancer or this cancer here, is by surgery and radiotherapy. Doctors know this very well, but students may not. You might ask me, so if there are two therapies, which one the doctors will choose? Basically, what happens for localized prostate cancer, which requires removal, surgery will be preferred. So there are people who are frail, who are really, really old, and they cannot undergo an invasive procedure, in which what will happen in those cases, those patients may be offered and will be offered radiotherapy. And this radiotherapy will destroy the tumor. Uh, with surgery, mostly it is radical prostatectomy, meaning the entire gland will be removed. The prostate cancer, which has gone into local invasion or metastasis, it will be treated by androgen deprivation therapy. Now, I was telling you yesterday, we work on a transcription factor called the androgen receptor. And basically, the goal of this therapy, androgen deprivation therapy, is to block the activity of androgen receptor. I'll again show you that slide on androgen receptor. Again, so what is happening here? We have drugs like enzalutamide, which directly block androgen receptor. And we give patients therapy called androgen deprivation therapy. The intention is same. You're trying to block the activity of a major transcription factor, which drives prostate cancer. So this therapy is commonly known as castration. Castration meaning cutting down the supply of androgens. This can be achieved chemically tissues in the bones, and in doing so, they actually dampen the tremendously the quality of life of a patient who has the advanced disease. And you can see the androgen receptor levels are going up. Like I was showing you the examples, have a look what happens when patients are being treated with androgen deprivation therapy, and what happens when androgen receptor becomes active in a cell. So this is what you're seeing is DNA inside the nucleus. This is a nuclear pore in a cartoon. This is androgen receptor, and it is bound by chaperone. And you can see there are at least 3 different domains. I don't want to overcomplicate, but these 3 domains are very important for androgen receptor to drive transcription. I told you day before yesterday that all transcription factors will need to interact with the DNA, and therefore they have something called the DNA binding domain. They may have different structures, but functionally their role is to allow the protein in which they're part of to take this protein to sit and bind to the DNA. But they have 2 other domains. One of them is called amino terminus domain, the N-terminus part of the protein, and they also have the ligand binding domain, which is the C-terminus part of the protein. In this cartoon, what you can see is that the androgen receptor is bound by chaperones. We know when androgen receptor is in a cytosol, it binds to chaperones, which stabilize the androgen receptor. We also call them heat shock proteins, the chaperones. Now, yesterday we were talking about a database of protein-protein interaction. That database that we discussed was called a string, if you remember. And we looked at what are the top binding partners of your favorite protein. We inputted androgen receptor and one of the proteins on top that has a score of 0.999, if you remember. That protein was none other than a chaperone. It was a heat shock protein. So androgen receptor binds to the chaperone when it is cytosolic. It is not active at the moment. So what is going to happen is that when the steroid hormone comes, it binds to the ligand binding domain, as is shown here. And the binding of the steroid hormone, androgens, testosterone, will cause this. They will bind to the protein. It will cause the receptor to change its structure so that chaperones can no longer bind to it. And what is now going to happen is that androgen receptor will be able to travel into the nucleus, binds as a dimer, which is not shown by the way here, and binding, it will also bind to some factors which we call androgen receptor interacting proteins. And what will happen, these are only few proteins shown. I was studying yesterday, we have identified over 1,000 proteins that bind to androgen receptor through our proteomic screen. So what is happening? You would ask me, hang on a minute. If androgen receptor is driving transcription, what is the role of RNA polymerase? Well, transcription factors actually create a platform for the transcription factor. RNA polymerase is a transcription factor. These factors, androgen receptor, for example, create an environment, create the conditions, create that very platform which we need for the RNA polymerase to bind to the DNA. And then RNA polymerase can drive transcription of target mRNA that they will want to target. One of the mRNAs which RNA polymerase regulates in this context codes for a gene, a protein called PSA, prostate specific antigen, which I was just mentioning a minute ago. So anything that you see in a patient blood PSA, the level of PSA reflects the level of androgen receptor activity. If you have high androgen receptor activity, you will have high levels of PSA. So what can you do in order to block this pathway, androgen receptor pathway, is to block the AR activation by the ligand. So you have these ligands binding to the androgen receptor. We have drugs. One of the drugs is called enzalutamide. And what you can do with these drugs when they're given as capsules, they block this ligand, the hormone, from binding to androgen receptor. And if these ligands cannot bind to androgen receptor, it comes back into the cytosol. It's not active anymore. And that is what is done using drugs like enzalutamide. This pathway becomes active. I hope this is crystal clear. One of the ways also that you can block androgen receptor activity is by blocking these proteins that bind to androgen receptor. If you can block these proteins that bind to androgen receptor in order to stabilize androgen receptor, you can actually decrease the activity of the androgen receptor. So either you can make new drugs that will block, that will compete with the ligand, that will compete with the androgen to bind to androgen receptor and activate it, or you can actually inhibit proteins that are part of the androgen receptor transcriptional complex. The major challenge is, first of all, is to identify which proteins bind to it. Unless you know which proteins bind to androgen receptor, you cannot think about decreasing the level of androgen receptor via interventions that target these proteins. I hope that is clear. So the proteins that bind to the androgen receptor, we call them androgen receptor associated factors, or ARFs, or sometimes we call them ARIPs, androgen receptor interacting proteins. That's one thing. Now, my laboratory works on both of these aspects. What we are trying to do in the laboratory is trying to develop novel drugs, novel AR inhibitors using approach one that will block the androgen receptor activity in tumors. The second thing that we are doing in the lab is to perturb the transcriptional complex of androgen receptor. If we make androgen receptor unable to interact with the DNA, it is not going to be able to drive transcription, is it? And as a result, the androgen receptor will become inactive. So these are the two approaches that my laboratory takes in the lab. To tell you more about in more detail, these are the three domains of androgen receptor, and you can check with your favorite protein of interest as well. If you are talking about the estrogen receptor that drives specific subtypes of breast cancer, essentially you will see these three domains, because both androgen receptor as well as the estrogen receptor are part of the same superfamily of nuclear receptors, nuclear hormone receptors, as we call them. So, in the androgen receptor, we know what DNA binding domain is doing. The function of DNA binding domain is to allow the protein to bind to DNA. But in androgen receptor, we have the amino terminus domain, which is the major transcriptional driver. Without this, androgen receptor will not be able to synthesize mRNA of the target genes. And then there is a second driver in the ligand binding domain. So when we are studying prostate cancer with an intention to develop drugs to block androgen receptor activity, we are trying to target driver 1 or driver 2. Now have a look what happens with driver 2. This is the driver that I was talking to you about, the ligand binding domain, which binds to testosterone. Individuals taking testosterone for muscle building, that's a very bad news for them, because this is something which can actually promote cancer diagnosis, taking excessive amounts of testosterone. And what happens? Binding of testosterone triggers the receptor activation. We discussed this on the previous slide. And once the receptor becomes active, it can actually cause prostate cancer. Not a good news. Well, we have very good drugs, thankfully, that can block the activation of the receptor called the bicalutamide, which is a generation 1, first generation anti-androgen. But we also have a newer drug called the enzalutamide. The functional analogy is the same. Both of them compete with androgens to bind to the ligand binding domain. This is how prostate cancer is being treated in the laboratories, along with androgen deprivation therapy is being treated in hospitals all across the globe. What we don't think about is targeting this driver, and there is a very good reason for not being able to target driver 1. The reason is that the driver 1, the structure, it does not really fold into proper 3-dimensional structures. It doesn't make those alpha helices or beta sheets. There is no defined structure. Unless you can define a structure of a protein, there is very little you can do in terms of thinking of blocking the function of protein. So you need a three-dimensional structure of a protein. Proteins don't look like they're shown on your screen. This is a simple, simplified cartoon of androgen receptor domains showing you the functional domains. So coming back to this, there is nothing you can do. In the human proteome, a substantial part of the proteome does not really fold into a proper structure. It gives them flexibility. Same is the case with, sadly, androgen receptor. The major driver of this disease, androgen receptor, is driven by driver 1 domain. And this domain is the major domain that drives the androgen receptor activity. And unfortunately, this domain is intrinsically disordered. It does not really fall into a proper structure, meaning it cannot be targeted by drugs. And in fact, we don't have any drugs at the moment which are used in the clinic which will cure prostate cancer by targeting driver 1. Most of the drugs, in fact 100% of the AR targeting drugs which are used in the clinic for the treatment of prostate cancer, they target the LBD or the driver 2. Hope that makes sense. So what happens to the resistance? Now this will bring you back to the lecture 1 and lecture 2, the first day lecture and second day lecture. How does the resistance develop? There is no new mechanism as such. Where there is something new, I'll tell you. Have a look what happens. When prostate cancer patients whose tumors are being driven by androgen receptor, they are treated with first generation drugs, first generation LBD antagonist or LBD inhibitors. We see the receptor mutates, and the mutation are not happening at protein level. As you know, they're happening at the level of DNA, specifically within the exons. And here what is happening, there are point mutations happening, and there are several point mutations that are known to happen within the ligand binding domain. As a result, what happens sometimes, the receptor in cancer cells becomes active despite being bound to the drug. So normally androgens binding to this receptor activated, anti-androgens or the AR inhibitors binding silence the function of the receptor. But because of these point mutations, the receptor is now becoming active by the very chemicals which were meant to stop this function. I hope that makes sense. So what would you call them, these mutations, as being? They're gain of function mutations. They've given them, the androgen receptor, ability to have an abnormal function that allows cancer to progress. Okay. We use these drugs. Hydroxyflutamide is one example. Cyproterone acetate, another example. Bicalutamide, another example. We use them for a very long time. 15 years ago, almost, we got new agents that can still target the ligand binding domain, but those new agents were active in androgen receptor mutant phenotype driven cancers. We call them second generation drugs, and these second generation molecules actually can inhibit cancer growth. They are saving lives. Few months to maybe a year, many patients actually get some relief from their cancer. Now you will ask me, so what is the problem? There are already second generation drugs. Well, as I said, and Dr. Aziz was just alluding to, there will always be resistance, sadly. And it did not take very long before the resistance emerged to second generation inhibitors. And you know what happened here? They had point mutations, but one of the most remarkable change that happened to them is that there was a loss in the exonic sequence which codes for ligand binding domain. So now these cells in prostate cancer, which became resistant to second generation drugs, like enzalutamide. So doctors are giving enzalutamide. They're all very happy. We have a drug, enzalutamide, that works. Yes, it does work in patients for a good few months. The patient is healthy. They look all right. And then they're measuring their PSA from their blood. And the PSA is dropping. PSA is one of the genes which is regulated by androgen receptor. So a doctor, by just taking some blood, can make predictions that actually you are now in remission. Your cancer is gone. Your cancer is shrinking. On scanners, it looks like the cancer volume is decreasing. The locally advanced disease, for example. But again, what happens? The PSA starts to increase upon subsequent blood test. Now, people don't understand what's happening. It turns out that the receptor has mutated again. And the receptor has mutated so that it does not really have the ligand binding domain anymore. But the major domain which makes the receptor active, the NTD, is still there, as you can see. So although the receptor has become shortened, any treatment with an intention to block ligand binding domain, including the androgen deprivation therapy, they're not going to work. Why? Because the receptor is now totally independent of the need of requiring an LBD. They don't have LBD. So if I show you some data taken from biopsies and androgen receptor level, expression level in IHC established, have a look. These are the patients. These are matched patients. So pre-development of resistance and post-development of resistance to second generation inhibitors. And have a look what is happening. There is a tremendous increase in the expression of this small truncated variant of androgen receptor in patients who have developed castration-resistant prostate cancer. This truncated form you do not see in patients who are still sensitive to castration or have a castration-sensitive prostate cancer. So nature is always trying to improvise and finding ways. The nature is more steps ahead than we are in finding novel ways to promote in this context the cancer. You can see here there are point mutations in response to first generation, and one of the most prominent change that happens that leads to resistance to second generation inhibitors is the loss of ligand binding domain. But some of them will also have the loss of the some of them will have intact ligand binding domain despite having resistance to second generation inhibitors. They may have some new point mutations. That is also known. Recent data came from France which shows epidemiological genomics data showing that patients who become resistant to second generation drugs and drugs like DNA damaging drugs like docetaxel have a specific point mutations in the ligand binding domain of androgen receptor. But notice one thing. These cancers still require androgen receptor. They are still dependent on androgen receptor, either full length or the small truncated isoforms of the androgen receptor. So when this came in last few years, we started to look at, okay, so what is happening when you don't have the ligand binding domain? I didn't have to go much further than looking at my own PhD thesis, which I completed in October 2006 in Germany. So as a PhD student, I was, of course, I had left India for the very first time. I started my PhD in October 2003 to 2006. And there's, of course, a lot of work in the new lab, lots of pressure as well to perform. I had left two JRFs, both CSI and ICMR. I had resigned basically, and there was no going back. So I started making these constructs of androgen receptor to see there are these domains, N-terminal domain, DNA binding domain, and ligand binding domain. What do they do? And I was studying proteins that bind to androgen receptor and seeing what is the impact of androgen receptor on androgen receptor of these proteins, those proteins bound to androgen receptor. So you see, I have been working in this area for a very long time. It actually enhanced the productivity, maintaining the same research area. I did go here and there, but most of my focus throughout my research life has been on prostate cancer and largely on androgen receptor. So coming back to what I was showing you, these small truncations that are generated in a cells that lack the ligand binding domain, which are generated, produced as a result of cancer becoming resistant. I had done some experiments where I had, as a PhD student, where I removed the ligand binding domain. And I made these constructs, and I was looking at the effect of these constructs on AR dependent, the transcriptional activity of androgen receptor. So this is a simple reporter assay, which can allow you to evaluate how active the receptor is. So on y-axis, if you see a larger bar, it simply means that the activity is higher. And you can see here without androgens, androgen receptor is not active. You see very little activity. But as soon as in your experiment, you treat your cells with androgens, you see a 50-fold receptor induction. Activity is gone up. I hope that is clear. Sometimes if you remove parts of the NTD, you see that the receptor activity is gone. Totally gone. So let's come to this point. I was making this. Those mutations had never been found up until that point. But I removed the LBD thinking that because it is a driver, we will lose the activity of the receptor. But actually something opposite happened. As opposed to here, where there was a 50-fold increase, we saw a 3-fold increase in the receptor activity. So now the receptor has become super active as a result of losing this driver. So basically, LBD is indeed a driver, but there are constraints. When I say driver, it simply means the LBD needs to be engaged with androgens to be brought into the nucleus to carry out this function. But what has been happening is this molecule here, this protein, is able to come into the nucleus anyway. And look at what is happening. The receptor is now super active in presence of not only androgens. Have a look, even without androgens, the receptor is almost as active as it is in the presence of androgen, meaning the receptor has become constitutively active. I was showing you that battery inverter alternator, the switch the other day, which has on both sides. Basically, this is a classical example. You have, by removing the LBD, created a molecule that does not really require this androgen receptor truncation, does not require any androgen to bind to it and activate it. It's become active all the time. Now, pharmaceutical companies, drug industries, doctors can treat patients who have these androgen receptor variants. They can treat them with anything that targets the LBD. They're not going to see any efficacy, would they? No, because the target domain is missing. Why nature is doing this, we came to know a very, in 2006, but those eventually became clinically relevant after more than 10 years, and we started to see this as a result of new generation AR inhibitor drugs, that these truncations can be generated in patients actually. So we were able, in this case at least, we were lucky enough to predict this before the resistance actually happened to this. So we modeled this resistance beforehand, as you can see through this work. So you can think this as a scenario where, in order to survive the attack from a predator, this lizard would shed their tail. And as you can see, this jumps and dance. And in doing so, the lizard is able to be fooled. They are able to trick the predator who will attack the tail. They again can grow tail. but they survive. And likely exactly something similar is happening here, where the androgen receptor, by removing one of the most important domain, survives any AR inhibitors which targets the LBD. So you can see here this receptor is now always active, even in the absence of androgen or in the presence of it. It always becomes active. Shall we take a 10 minutes break, Dr. Aziz? Okay, I think we will stop. Okay, perfect. I'll see you soon. If you have any questions, please place them in the chat box. Great. We can start now. Thank you. So, welcome back, everyone. What we were discussing before we left is how this protein that is a major driver of prostate cancer, the androgen receptor, can become active despite the treatments intended to stop its function. And one of the ways it does so is by through mutations, which allow the androgen receptor to lose its ligand binding domain. And in doing so, the androgen receptor becomes super active, as you can see here. You can see here, this is an example I gave, and this is basically showing you the constitutive activation of the receptor. So it took many years afterwards to start defining what are the characteristic features of androgen receptor variants. These are smaller truncations of the androgen receptor I showed you. And these two papers come out. You can see here, one of them is in New England Journal of Medicine, which actually showed that having the presence of androgen receptor variant, one of the variants they study, which is one of the most common ones for the androgen receptor variant 7, leads to resistance to AR targeting drugs like enzalutamide and like the abiraterone. So this androgen receptor variant presents can serve as a biomarker for resistant for clinicians to make sure that the treatment that they're giving may not actually work intended to target the ligand binding domain. But then the good news is that there was another paper which came out from Memorial Sloan Kettering from Sawyer's lab, which showed that actually the androgen receptor variants do not function alone. Remember, I was telling you androgen receptor binds to DNA as a homodimer. It turned out that androgen receptor can also bind to DNA as a heterodimer, meaning androgen receptor full length and androgen receptor variants, the smaller form, can actually bind together and then sit on the DNA to drive transcription and can lead to expression of genes. So what we ask is it possible that we can target both of them at the same time? Of course, androgen receptor, if you have a look at the structure, the androgen receptor full length has both driver domains intact, meaning the NTD, and the ligand binding domain. But the variant, the androgen receptor variant, the smaller, shorter truncation, does not have the major driver, which is a drug target. And sadly, targeting NTD has not been very successful to date. So why we are targeting androgen receptor signaling using AR inhibitors? Because as you see, this is showing you the drug use in a US prostate cancer drug market. And as you can see in these bars, these are different years, and you can see currently we are here. You can see most of the market is dominated by prostate cancer treatment is dominated by hormone therapy. Hormone therapy is nothing but the AR inhibitors that are used in the clinic for the treatment of prostate cancer. How big is the market? You can see over 3 billion US dollars. Some of this market is also covered by chemotherapy and immunotherapy and other target therapy. So, but most of the time you can see here the androgen receptor targeting strategy remains to be the favorite one for clinicians as well as for drug industries, and therefore there are efforts to develop better drugs. And you can see here the use of androgen receptor targeted therapy is only going to increase in the future. So what we are doing in my lab to support this targeting of androgen receptor novel agents, especially the variants and androgen receptor full length, is shown in this paper, where what we showed is that We can combine two drugs. One of them is enzalutamide, a drug that targets the ligand binding domain, and there is an this drug, other drug. This is called EPI-001. This drug was in clinical trials, but since the clinical trials have stopped, they did not show any benefit. But it was a good tool compound to use in our experiments because of the fact that it can inhibit in culture dishes the NTD activity. This was from NASA Pharmaceuticals. And we combine this drug with enzalutamide that targets the ligand binding domain using a linker. And what we could establish is that this drug, of course, this is a big molecule, will have lots of issues if given to humans or mice. But in cell culture dishes, we could see that this drug was much better at blocking the androgen receptor activity and therefore the growth of cells, because the prostate cancer cells in which we did this experiment, they were reliant, they were dependent on androgen receptor. So by doing this work, this research, we showed that actually gave the proof of concept that you can target the NTD and LBD together. And if you do that, then you will have a better killing and you can actually circumvent, you can stop the resistance from happening. So that was the initial work. Since then, and this work is published, as you can see, but since then we have developed better inhibitors. Please this note that this is unpublished, and please make sure that this is not something you post on social media. So what you're seeing here is we use this compound enzalutamide, and we did structural activity relationships analysis and generated over 100 derivatives and identified this molecule. So this work is in the chemistry part is in collaboration with the Department of Chemistry, University of Cambridge. And you can see we made this drug duotamide. So if you Google this drug, you will not find anything at the moment. But basically, duotamide is a dual androgen receptor targeting derivative of enzalutamide, hence the name duotamide. And duotamide is, as you can see, it's a small molecule, and duotamide actually is capable, as you can see here, is able to bind. This is a FRET assay. Those of you who do biophysical analysis, biophysical assays, would know what this assay is. But basically, what you're looking at is the competition, is the binding of duotamide to the ligand binding domain. And we are comparing it against other androgen receptor inhibitors, but also androgen receptor agonist or androgen receptor ligand, the androgen or R1881. And you can see in this experiment something very clearly. The androgen receptor ligand, the R1881, this binds to androgen receptor very, very quickly, and in doing so, it replaces a labeled ligand. And so androgen receptor in this all of these settings has a very strong fluorescent ligand bound to it that gives a very high polarization, uh, as shown here. But as you have anything that binds to androgen receptor which is not labeled, the label will start to go away. Who is best at it? The hormone, the androgen itself, the R1881. Now, if you compare this with duotamide or enzalutamide or bicalutamide, all three drugs are actually quite good in inhibiting the androgen receptor activity by binding to the ligand binding domain. What you can also notice is that this EPI drug, which targets, as I mentioned, the NTD, does not bind to the ligand binding domain. So we could establish that duotamide actually works like its parental drug. How? Because it can bind to the ligand binding domain. But also what we did is this biophysical assay called the SPR, or surface plasmon resonance. And what happens if you see the binding of the protein, the recombinant protein with the molecule, you would see a peak. And exactly this is the peak we saw when we used duotamide, but not in the negative control, not with the buffer alone. So we got an idea that actually duotamide binds to the androgen receptor NTD. So now, in addition to having a big molecule, which was a tool compound, which we used and proved that it can target both domains, we have a much smaller intact molecule which can target both the LBD and the NTD. So we use this in a reporter essay. I was showing you some reporter essays previously from my thesis. Have a look at this reporter essay. If we do a reporter essay to look at the transcriptional activation ability of androgen receptor, not full length this time, only the smaller truncation, the variant NTD-DBD. Notice the LBD is missing. What happens here if we use enzalutamide in increasing doses? You can see the doses are incremental. Do you see any decrease in the receptor activity? I don't. Meaning enzalutamide does not and cannot target this receptor. Why? Because the LBD is missing. Now we have drugs. Two of them were in clinical trials. This drug here, mesofenitin, from ESA Pharmaceuticals, and this drug here from Oncternal Therapeutics. Both are US-based companies targeting the androgen receptor N-terminal domain. Do you see any inhibition of the androgen receptor amino terminus domain or this smaller truncation variant? I don't see any. Then one of the postdocs in the lab did some experiments and showed that duotamide at increasing doses impressively inhibit the amino terminus domain of the androgen receptor as shown here. So in cancer cells which are driven, whose growth is driven by these smaller truncations, the androgen receptor is still active and it is driving the inhibition. The duotamide is driving the inhibition of it. So because it is a target, any cell that harbors it depends on it. You block the androgen receptor activity, those cells, what should happen to them? They should die. And we tested that. Have a look here what happens. These are some in vitro experiments and some tumor xenograft experiments we have done. Have a look what happens. These are 4 cell types, 1, 2, 3, 4. I don't want you to go into the details of the names, but have a look. This cell is driven by a full-length androgen receptor. This cell type has both full-length and the variant. These 2 cell types only have the variant. And then we tested 2 compounds, an LBD targeting drug enzalutamide, which is a clinical use standard of care for prostate cancer, or we tested duotamide. Do you see what happens here? With duotamide, you get a much better cell killing at the same dose compared to enzalutamide, which does not give that great inhibition of cell growth. And you see very similar things here in other cell types, which only express variant. So this is the sort of experiment that one would do to establish the efficacy of drugs. These are cell growth curves, by the way. What you are seeing in this tumor xenografts, that the tumors were established, shown in blue here. In this animal, you can see here, we treated it with a single dose of the drug. And you can see here what is happening. These cells now express both the full-length expressing cells as well as the androgen receptor truncation expressing cells. Both of them have heightened increased level of cleavage caspase 3, which is a biomarker for a cell death. So you're able, within this short window, as little as 24 hours, you're able to trigger apoptosis by duotamide in tumors, not the cells growing in a culture plate. So you will ask me, hang on a minute. This is the drug that you're developing, duotamide. These drugs have been in clinical trials, and you showed in your experiments they don't really work by inhibiting the shorter isoforms through this reporter assay. So what happened to these drugs? Have a look their fate. So one of them in the same family, the EPI 506 compound, they stopped because of the high pill burden. The patients were taking 9 grams of the drug 500 milligram capsule, 18 capsules every day. This trial was terminated. You can check the trial currently available data through this identifier on clinicaltrials.gov website. And we in our hands, the reporter has said we did not see any changes in the reporter activity whatsoever. Then there is this another drug that we saw, Oncternal Therapeutics drug. We did not see any reduction in the reporter activity or the receptor activity, transcriptional activity. What happened to that? Well, last year or in 2024, they announced that their drug, ONCT 534, the exact molecule I showed you, this one stopped to have any clinical benefit in treating patients who have castration-resistant prostate cancer. So both of them actually failed. So with duotamide, what we have found is a drug which can actually block both drivers, the driver LBD, which is a conventional driver, and the drugs used are the ones that are intended to target this driver. But we also have drugs like we also have duotamide, which can also block driver one, which is the NTD. Any questions? Let's open floor. We'll take some questions and then we will move on. If you want, I can